Hello, everyone. Today I'm going to explore how different impulsive compulsive disorders influence decision-making processes and which forms of treatment affect reward circuits through the lens of cognitive psychology by examining two research articles. So, the first article that we will be examining is titled Decision Making in Frontal Parietal Resting State Functional Connectivity Among Impulsive Compulsive Diagnoses. So, the authors of this article were motivated to explore decision-making profiles in individuals with spectrum impulsive disorders. They aim to understand how these profiles might differ between clinical and healthy populations and whether resting state functional connectivity in the frontal parietal network could serve as a biomarker for decision-making patterns. So, resting state functional connectivity, because it's a long word from now on, we're going to call it RSCF, refers to the measurement of how strongly different regions within the frontal parietal network of the brain are correlated with each other in terms of their neural activity. The study addressed theories related to dimensions in psychopathology, specifically focusing on how decision-making varies across different diagnostic groups and how brain connectivity might predict these patterns. So they tested whether decision making is shaped more by how often punishment happens than by long-term rewards. It also looked at whether decision making styles match diagnostic categories and whether unique brain connectivity patterns in the frontal parietal network relate to these styles. So a total of 114 participants were split into healthy groups and into healthy controls, OCD and ADHD groups. They completed clinical interviews, rating scales, but more importantly, two main tasks. The Iowa Gambling Tasks, IGT, to measure decision making, and the FNIRS to track brain connectivity. So the Iowa Gambling Tasks involves picking from 4 decks of cards to maximize profit, with each deck having different reward and punishment patterns. This study explored how diagnostic groups, resting state functional activity, affect decision-making and cognitive processes in OCD and ADHD patients to better understand how clinical conditions impact cognitive processes, specifically decision-making and functional brain connectivity. So the study found distinct clusters in participants' deck choices, showing how these patterns varied with diagnostic groups and demographics. D learners favored deck B, B explorers stuck with deck B, while the scattering group had no clear deck preference, setting them apart. There were no significant differences in deck choices based on diagnostic or sex, and in tables 2 and 3, confirmed that cluster membership wasn't linked to diagnostic labels or factors like sex, income, or education. Unlike the other clusters, scattering participants showed random inconsistent strategies, suggesting that task behavior depends more on individual traits than on clinical conditions. So, this indicates that while clusters exhibited clear patterns in the deck preference, diagnostic groups did not appear to influence these preferences. This suggests that behavior in the task may be more influenced by individual tendencies rather than specific diagnostic conditions. So, this highlights that people approach decision-making tasks, like the Iowa gambling task, in different ways. For example, some may prefer less profitable options, like in the activity, people chose deck B due to factors like loss aversion. Interesting, though, there were no major differences in the Iowa gambling task performance between diagnostic and healthy controls, which that individual differences within those groups seem to play a bigger role, challenging past assumptions about decision-making deficits in clinical populations. Another key finding was that the RSFC didn't correlate with the IGT performance. The author suggests that the frontal parietal network might play a more flexible, context-dependent role, that and that its connection to decision making should be studied alongside other brain networks. So overall, this study highlights the individuality of decision-making processes, suggesting that clinical diagnoses should not be solely relied upon to predict decision-making behaviors, which also calls for a more broader, nuanced approach to decision-making styles in both healthy and clinical populations. So the next article dealt with stimulant treatment. So ADHD is often linked to altered reward sensitivity, with studies pointing to dopamine dysfunction in reward-related brain areas, affecting impulsivity and inattention. While stimulant medications like lisdexamfetamine demesylate, from now on we are going to call that LDX, improve symptoms, their long-term impact on reward processing is less understood. Hence why the authors went through with this study. So they hypothesized that LDX, which is a stimulant medication, would boost reward-related brain activity, especially in the striatum and frontal cortex, which are key areas for reward and motivation. They also predicted that greater brain activity in these regions would correlate with symptom improvement, suggesting that stimulants help normalize reward circuit function in ADHD. So the experiment had two key phases. There was the choice phase and the feedback phase. So participants chose whether to approach or avoid visual stimuli based on expected outcomes during the choice phase, and they received feedback in the form of reward or punishment based on their choices. They were also presented with fMRI scans to track their brain activity, where participants completed a passive avoidance task. They were shown one of four shapes per trial, two linked to rewards and two to punishments. Responding to a reward shape earned in a virtual gain, while the other responding to a punishment led to a loss. And not responding at all resulted in no feedback. So this setup helped researchers study how participants learned to seek rewards and avoid punishments, along with how the LDX treatment impacted their decision making and learning. So participants underwent two fMRI scans, one after a 4-week on-drug phase and the other during a placebo phase. And ADHD symptoms were also tracked using the ADHD rating scale. The PA task measured reward-related learning and brain imaging focused on areas like the striatum and frontal cortex, which are all tied to reinforcement and reward processing. So researchers examined whether LDX increased brain activation in these pathways and whether this correlated with symptom improvement. So the study found that LDX did significantly reduce symptoms of ADHD compared to the placebo, as shown by lower ADHD rating scales on the on-drug condition. And these improvements were statistically significant with no carryover effects between placebo effects. So in Table 1 and Figure 2, you can see that these results are highlighted. So ADHD symptoms dropped significantly from baseline to on drug, and Figure 2's bar graph clearly showed a marked reduction in symptoms under the LDX compared to placebo with a p-value of less than 0.05. So the functional image findings show that brain scans revealed a positive link between symptom improvement and increased activity in the left caudate and right inferior parietal cortex, which are both areas involved in reward processing and attention. So this strongly suggests that LDX treatment enhances activity in these regions, potentially contributing to symptom relief. So the authors conclude that LDX modulates brain activity, brain regions involved in reward processing, which is linked to ADHD symptoms improvement, and significant increases in activation were observed in the putamen, caudate, and frontal cortices, supporting the idea that stimulant medications, which has been an ongoing debate, do enhance activity in reward-related areas to improve symptoms. So they highlight that increased brain activation during tasks involving decision-making, reward, and punishment feedback correlated with better attention and reduced ADHD symptoms. For instance, greater frontal cortex activation suggests that LDX helps with sustained attention in which is a core ADHD challenge. So some considerations to take into account was that the authors had a small sample size, which affected their ability to explore differences by sex or ADHD subtype specifically. They also didn't have a traditional control group, which may affect the clarity of results. And there was no significant changes in task performance, indicating that LDX's effects might be more apparent at the neural level. Despite these limitations, though, the study contributes to understanding how stimulants work, showing that treatments like LDX improve symptoms of ADHD by enhancing brain activity in regions tied to reward, attention, and motor control. How the authors do call for larger studies to explore these effects further, particularly across different ADHD subtypes. Thank you so much, and these are my references.